Hi everyone, my name is Felipe Vaz Peres. I am undergraduate student in biotechnology, and today I'm gonna talking about ContFree-NGS, a simple software that enables the user to remove contaminated sequences from their next generation sequencing dataset. So to start this presentation, I would like to introduce the problem of contamination in public datasets. So as defined by NCBI, a contaminating sequence is one that does not faithfully represent the genetic information from the biological source organism because it contains one or more sequence segments of foreign origin, and they could cause several problems in downstream analysis. The primary consequences of contamination are time and effort wasted on meaningless analysis, erroneous conclusions drawn about the biological significance of the sequence, disassembly of sequence contigs and false sequence clustering, delay in the release of the sequence in public databases, and the pollution of public databases. So, ContFree-NGS was developed to solve a problem I faced when I was working with sugarcane RNA-seq datasets. So, as you can see here, that is the taxonomic information of the contaminating sequences in the sugarcane datasets that I was working with. So here you can see the number of reads and percentage of contamination. So Acinetobacter baumannii is a bacteria, and it have almost 3% of reads in this dataset. So I tried to remove these sequences of contaminants with some softwares, and recently some tools have been made available that aim to remove sequences from contaminated organisms in next generation sequencing datasets, such as the Contaminer, a tool to unravel the presence of contaminating sequences in the set of reads that do not map to a reference genome, and also like Contaminator, a software that removes contaminating sequences from contigs, exploiting a taxonomic assignment file, and the QC Blind, an automatic tool to do unsupervised assembly and contig binding to identify and remove putative contaminants. But all of these tools have in common that they either require a reference genome of the source organism or need to perform assembly prior contaminant detection. So our goal with ContFree-NGS was to develop a simpler tool to remove contaminated sequences directly from unassembled reads without mapping and exploiting fast camera analysis implemented in taxonomic assignment engines commonly used in metagenomics, such as Kaiju, a fast and sensitive taxonomic classification software for metagenomics, and Kraken 2, a taxonomic sequence classification system. So here you can see our approach. ContFree-NGS was implemented as a single Python script using the BioPython module and the Python environment for tree exploration. And in order to access the contamination, ContFree-NGS exploits a taxonomic assignment file containing the region identifier and the NCBI taxonomic identifier for every sequence in the dataset. So here we can see the workflow of ContFree-NGS. So the first step is to have a dataset in FASTA or FASTQ format. Then you have to generate a taxonomic classification file, and with this taxonomic classification file, can run ContFree-NGS. The user must insert the target taxon, so all of the descendants of the target taxon will be maintained in the final filtered sequences, but I will talk about these parts later. So here, the first step is the taxonomic classification, and as ContFree-NGS exploits the results from a taxonomic assignment engine, users must use the proper switches to achieve an accurate classification. For instance, a proper value of the confidence switch in cracking true. So to perform the taxonomic classification with cracking true, we built a custom database containing the following reference libraries. So we have the reference libraries from archaea, bacteria, viral, human, fungi, plant, protozoa, and the NCBI non-redundant nucleotide database. And for example, this is one of the Kraken output. So here in the first column, we have the letter C. That means this read was classified by Kraken. The second column, we have the number, the region identifier. So here is the region 001, and then is the region 002. And in the third column, we have the NCBI taxonomic identifier for this region. So this identifier means that this region is from village plant. So with the taxonomic classification file in hands, the first step of ContFree-NGS is to create a SQL database to store the sequence index to make the contaminant removal process faster. And this is done using the biosec.io index db function from BioPython. And ContFree-NGS was the first software that I developed. So in this step, I tested various ways to do this process faster. And finally, in the contamination removal, ContFree-NGS creates a list with the NCBI identifier for the target taxon and all the identifiers of its descendants according to NCBI taxonomic database. So, for example, in this step, NCBI creates a list with the identifier of the target taxon. So, if village plant is the target taxon, that is the ID, and all its descendants, I mean all the descendants of village plant is included in this list. So it so this ID is from sugarcane. Then ContFree-NGS iterates over the taxonomic assignment file, and if the read was not assigned to any taxa, it is saved in a FASTQ file for unclassified reads. You can see it here. And if the read was assigned, it will check if its taxon is found in the list of target taxon descendants. If so, it will save the read to a FASTQ file for filtered reads. Otherwise, the read will be discarded. So this is done here in this step of contamination removal. To evaluate ContFree-NGS, we run ContFree-NGS against 3 sugarcane artificially contaminated datasets. So the dataset A contains 50,000 of reads, dataset 2 contains 250,000 of reads, and the dataset C contains 1,250,000 paired end reads. So in all these datasets, 80% of the reads came from sugarcane, 15% of the reads came from the bacteria Acinetobacter baumannii, and 5% came from the fungus Aspergillus fumigatus. So here is the representation of the taxons in these datasets. And after running Kraken 2 with confidence 0.05, the following sequences was classified in some taxon. And as you can see here, for the three datasets, nearly half of all sequences were assigned to a taxon. So we can choose if we will remove these reads or don't. So in the final step of the evaluation, we looked at the numbers of sequences in the datasets. So that is the distribution before the contamination removal, and after the contamination removal, selecting the plant taxon as the target taxa, we can remove all the contamination with ContFree-NGS. And that is the run usage and time consuming of ContFree-NGS. And as you can see here, the wall time scales fast with the number of the reads, but the memory is constant. And as I mentioned earlier, ContFree-NGS is the first software that I developed. So I tested various ways to do this process faster. And here you can see the comparison of the versions 1, 2, 3, and 4. And that is the runtime and run usage for filter 10,000 reads. And as you can see here, in the version 1 and the version 2, it costs 11 minutes to filter 10,000 reads. And in the versions 3 and the versions 4, we have only 4 and 3 minutes. And that's because in these two versions, the version 1 and the version 2, I stored the taxonomic identification and the region identification in a Python dictionary, and this Python dictionary was very large, and this is very time consuming to do this removal. And with the SQL database, this process this process is a lot faster. And the same happens when I tried to filter 50,000 reads. So here we can see the time consuming and the run usage for the version 1, 2, and 3, and 4. And for the conclusions, ContFree-NGS is a very simple filter and useful tool that removes sequences from contaminating organisms in NGS datasets. Memory usage is low and independent of the number of classified sequences, and the wall time scales rapidly with the number of classified sequences. And as ContFree-NGS exploits the results from a taxonomic assignment engine, users must use the proper suite to achieve an accurate taxonomic classification. And that's all. Thanks, and feel free to ask if you have any questions or suggestions.